Hello student, this is biology lesson for grade 12. Lesson 3. The main content of this lesson, lipid, we will see protein again. At the end of this lesson, student, you will be able to list and describe structure and the function of lipid and the protein. Student, do you remember what we have seen in our last lesson? Well, we have seen water and the carbohydrate. Nowadays, we are going to see lipid in the protein. Student, do you know what this lipid mean? How lipid can be differentiated from carbohydrate? What may be some advantage of lipid? Well, nearly all lipid can be made from element carbon, hydrogen, and that of oxygen, just like that of carbohydrates. But the proportion of oxygen here is less than that of carbohydrates. Lipid is insoluble in water, but it can be soluble in some nonpolar molecule like ether, like chlorophyll, and that of alcohol. Lipid is made from its respective monomer, fatty acid and alcohol, but it is not a polymer, but micromolecule, because its building block is not similar. Lipid are triglyceride, which is formed when one glycerol and 3 fatty acid molecule become esterified or bonded together. Phospholipid, which is formed when one glycerol, 2 fatty acid, and the phosphate group become esterified together. Else, waxes, which is made from that of fatty acid and that of long chain of alcohol bonded together. Do you know where wax is found? Well, the lipid found on the surface of bird's feather. It is waxes. And some type of lipid which is unusual lipid, sphingolipid, which is a nitrogen-containing lipid found in myelin sheaths, found surrounding a nerve cell, is 2 lipid. So why we say nearly all? Because there is some nerve cell that contain lipid, there is a nitrogen element may be contained there. Well, let's see the structure of triglyceride. Triglyceride is an important lipid which is formed when one glycerol, alcohol part, and again 3 fatty acid, long chain of hydrocarbon, and acidic part become joined together. So here there is carboxyl functional group is found here. There is 3 fatty acid that can be joined together by ester bond to form triglyceride. Triglyceride is very important in releasing energy during the respiratory process. So it is considered as a respiratory substrate just like that of carbohydrates to release energy. Notice this one, it can act as a thermal insulation, temperature insulation. An adipose tissue that is found below the animal skin is very important for this activity. Another, it is very important in buoyancy, animal to become bounce in water. By reducing the density of animal, allow animal to become bounce on the surface of water. Another, it is very important in waterproofing purpose. Oil secreted on the surface of animal feather or that of animal skin, proof as it is a lipid or triglyceride. So triglyceride serves this and that of this life function as an energy, and again temperature regulation, buoyancy activity, ensuring presence of lipid or not can be ensured here. It is very important. I told you that one important component of lipid is fatty acid, which is by covalently bonding of hydrocarbon, long chain of hydrocarbon covalently bonded together to form this functional structure, which you call carboxyl group. If you ask it what is a functional group that lipid can be shared with another carbohydrate, especially protein is, they can share carboxyl group. This can dissociate into that of carboxyl and that of hydrogen ion, making this fatty acid is acidic solution. Fatty acid may be saturated, monounsaturated, and polyunsaturated based on the presence or absence of double bond. If fatty acid is totally long chain of hydrocarbon joined together by single bond, we call they are saturated. No double bond presence. But here, if only one double bond found, we can say it is said to be monounsaturated. And again, if more than one double bond is found, we say it is said to be polyunsaturated. The presence of polyunsaturation in our membrane especially and in our daily diet is very important in determining cholesterol accumulation. It is very important. So, including some polyunsaturated, just like that of margarine, that of oils, is very important because they are rich of polyunsaturated. Well done, student. Another very important type of lipid is phospholipid. Student, do you know what by means phospholipid mean? And how we relate phospholipid with this structure? Well done. Phospholipid as another lipid, especially triglyceride, it is made from that of 2 fatty acid and again 1 glycerol and again 1 phosphate. Phosphate when they are joined together by special phosphodiester bond here. There is a linkage, 1 fatty acid removed here and again the phosphate become added. This makes phospholipid. Phospholipid become organized into 2 distinct regions. The one region is said to be hydrophilic region. It is considered as a water-love heading region which contain a phosphate part, a water-loving part. It is a head section of a phospholipid which can water-loving, considered as water-loving. Again, it can include a phosphate part here. Hydrophobic region. This is a water-hating region that contains a long chain of hydrocarbon tail that faces inward away from the water. This one is outward into the water, inward away from that water. Water-hating hydrocarbon tail. Whereas hydrophobic is water-loving phosphate head they are. This dual hydrophilic nature of phospholipid is very important in making membrane to become arranged in bilayer. So, phospholipid is the most important component of membrane bilayer. There is a hydrophobic region which contain hydrocarbon tail that move inwards away from the water, whereas there is hydrophilic head, a water-loving phosphate group that move outward into that of the water. This dual nature of phospholipid, hydrophilic, hydrophobic phospholipid, is very important to make membrane to become arranged that of bilayer. So, phospholipid is the key for that of a membrane bilayer formation. Good. The another topic that we are going to see in this lesson is protein. Do you know how protein can be similar with that, the earlier lipid and again carbohydrates? Well, they share so many things together. And again, they maybe share so many functions together. When we say protein, protein is very important in the complex than that of the earlier. That include element carbon, hydrogen, and the oxygen. Additionally, nitrogen and the sulfur is present. And again, Protein, just like that of lipid, it is very important part of plasma membrane. For example, lipoprotein, transporter protein, carrier protein, channel protein. They are a part of protein that found in membrane. Notice this one. Protein is very important in enzyme catalytic activity because enzymes are made from that of a globular or ball-like protein. Another, protein is very important in transportation of materials through the cell. For example, hemoglobin is very important part of which is to transport oxygen across the blood. Another, protein is very important in immune system. When antibody become recognized antigen, recognition of antigen in the antibody each other, which is a protein again. Protein is very important as a part of chromosome. When DNA histone protein complex to form a chromosome here. And again, protein is very important as a chemical messenger molecule, where the hormone that is found in living organism is very important in recognition, and again in recepting to certain part, to have certain function in transmitting of message, neurotransmitter especially, chemical messenger, hormone. So protein is too complex with a diverse function for the life. It is better to remember what function protein can be served for the life. So, well done. Protein, as the earlier carbohydrates, it is made from that of two functional group. We say that monosaccharides are made from that of two functional group, aldose and that of ketose part. Whereas these two made from that of two functional group again. There is a basic functional group, amino parts, and again acidic functional group, carboxyl parts. I told you before, as lipid contain fatty acid, which is a carboxyl part, acidic. So lipid in the protein share together carboxyl functional group. Protein as another micromolecules or that of a polymer, it is made from that of amino acid, a smaller building block called amino acid. When they are joined together by special condensation reaction called peptide bond, there is a peptide bond linkage to form a protein here. There is water releasing here. Water can be released during the process. This takes place when hydrogen atom lost from that of the amine group here, and again OH group lost from that of carboxyl group, releasing water in the forming amino acid. When so many amino acids become joined together, joined by that of a peptide bond, they can form is polypeptide. A bond, a union of many peptide bond that contain amino acid can form is peptide, polypeptide. So protein is made from that of 20 amino acids. So, well, amino acid have its own basic structure. All 20 amino acids that is found in living organism, they have the same basic structure. That all structure become attached around the alpha carbon. These are hydrogen atom. These are carboxyl group. These are amine part, and again R functional group or R side chain. Student, do you know what is the importance of this R side chain in amino acid? Good. Each 20 amino acid have their own unique R side chain, which means very important in determining the property of all amino acid, whether they are acidic part, whether they are basic part, whether they are non-polar, whether they are polar, soluble in water, and the insoluble can be determined by this side chain. Side chain is very important. So each 20 amino acid have their own unique side chain. So remember, these 4 parts are that come together to form amino acid structure. Good. Protein can have about 4 main levels. These 4 main levels can be started from that of primary structure. When we say primary structure, it is simple and that of long linear chain of amino acid that can be joined together by peptide bond here. We call primary structure of amino acid it is amino acid sequence by itself when they are linearly linked together by peptide bond. There is a peptide linkage here. Peptide bond linkage together. Here, each 20 amino acid in human body can be arranged in different way that determine what type of structure in the function each protein have. And again, what is the position in the type of amino acid in polypeptide chain R is determined by amino acid sequence, where this amino acid sequence in the cell can be determined by the gene. It determines the gene. These genes that are a part of nucleotide, a sequence of bases, can be determined the structure. Structures in the function they can be performed, and the position is the type of amino acid in polypeptide chain can be determined by the sequence of this gene. Well done, student. So, for example, this is amino acid sequence, which is called primary structure. A primary structure of amino acid here. For example, insulin, which have about 51 amino acid sequence. Secondary structure. Student, do you know how secondary structure formed? Well, secondary structure of amino acid formed when the primary structure amino acid sequence become bonded together by this bond between the carboxyl acidic part and the amino group or basic part, they form this secondary structure. This bonding may be when they become tends to pull towards in coil manner, they can form alpha helix, a protein that is found in hair and another body structure. And again, if they can be linked in parallel manner together, they can form a sheeted or that of plated manner, which is said to be beta plated sheet. So, a protein found in our nail, a protein that is found in our hair, and a protein that is found in a horn, a protein that is found in a feather are some part of secondary structure. This contain, if they are arranged in plated manner or parallel arrangement, they form beta sheets. If they are tends toward this form hydrogen bond become tends together in coiled manner, they can form this alpha helix. So, alpha helix in the beta plate are the two most important example of secondary structure of amino acid. Tertiary structure. Tertiary, as its name indicates, when secondary structure become folded together, further folding of secondary structure become make tertiary structure. It gives unique three-dimensional shape for that of protein to perform specific function here. Unique three-dimensional, unique 3D, unique three-dimensional structure can be found here. As a result, they can capable to perform a specific function. This is because of tertiary structure. For example, enzymes are specific. Enzyme, which is a globular protein, are specific to catalyze specific chemical reaction. As a result of this one, the shape of or 3D of their active site enables them to become bind to that of a specific substrate only. Enzyme catalytic activity to catalyze specific chemical reaction because of the shape of their active sites. Good student. Another, a hormone or a messenger is targeted to receive to that of certain specific messenger because of their structure too. They can receive to that of a specific, a hormone that's found on the membrane can be targeted to that of a specific messenger only. Antibody can be targeted to that of specific antigen. They can recognize, they can differentiate an antigen, specific antigen only. In immune system, in body defense system, they can capable to identify or recognize specific antigen only. These three examples are the most important example of tertiary structures. When secondary structure become folded together, they form a tertiary structure, which is very important with that of 3D or three-dimensional shape to give specific function. Student, I told you that primary structure, which is amino acid sequence, joined together by peptide bond. Secondary structure by hydrogen bond, where tertiary structure can be joined by the following three main bonds. There is hydrogen bond too between R group of amino acids, and again disulfide bond between R group that contain sulfur. Hydrogen bond may be found here. The one that takes place between positively charged R group and that of negatively charged R group. There is hydrophobic interaction too. A reaction between that of hydrophobic R side chain too. So they have specific bonds that they can hold them together. The final, most complex, and the further folding of the earlier protein become forms this quaternary structure. Is most complex than that of the earlier type of protein structure. This means formed when two or more polypeptide chain of the same amino acid subunits joined together to form a functional group, to form a functional protein here. So this is bit complex than that of the earlier. For example, hemoglobin, which is an oxygen-carrying pigment in red blood cell, which is made of 2 alpha subunits and 2 beta subunits. Collagen, which is very important quaternary structure, which is found in tissue of many mammals, is some example of quaternary structure. Notice this one. Based on their molecular structure, amino acid can be classified into 2 main parts. These are fibrous protein, which have a long fiber-like structure. It resembles tertiary structure with a fiber. There is a fiber-like structure. For example, collagen in animal tissue, and again keratin. And again, the else part of amino acid based on their molecular shape or structure is globular protein, which resemble tertiary structure, having a ball-like structure, globule or that of ball-like structure. For example, enzyme, which is 3D structure, receptor protein, which is hormone, is also a ball-like structure. So, student, generally, in this lesson, we have seen lipid and some part of lipid with their specific function. And again, we have seen protein component, its function, its functional group. And again, we have seen some structure of protein, like primary structure, which is amino acid sequence held by peptide bond. Secondary structure, which is joined together by hydrogen bond. Tertiary structure and the quaternary structure. And again, we have seen protein to be classified as fibrous protein and again globular protein based on their molecular structure. In our coming lesson, we will see enzyme. For now, This is all about this today's lesson.